Hello, back to continue our discussion on bacteria. So we left off looking at the general overview, we should say like the introduction to the prokaryotic cells, the idea that bacteria are ubiquitous, that they're not all pathogenic, they're not all harmful, they are some very beneficial species. We look at generation times. We're going to continue on now looking at the idea of bacterial taxonomy. So how would the scientist, the researcher, you or me, be able to identify what type of species we're looking at, right? So yes, we know there's some good ones, some bad ones, but how would we know actually what species we're talking about? So this is Biology 1307. We're going to get some introductory information. We're not going to dive in as deep as we would if we were in let's say a microbiology class, microorganisms in disease class. So this is again just a generic overview, a little slight intro into the taxonomy of bacteria. So I think you were doing some lab stir assignments with the gram stain technique. Well, before we can get to gram staining, we first have to culture bacteria. So the way that would be done, well, we would get a clean agar plate, and depending on what type of funding we have or what type of bacteria we're trying to grow, there's different types of growth media. But growth media is basically like a mixture of nutrients and amino acids and minerals that these bacteria would need to grow. So here's just the regular agar. Here we call this blood agar. There's chocolate agars. There's different types of growth media. Right? Think of like plants. There's different qualities of soil for different species of plants. It's the same kind of concept here. But when they start out, these plaques, or I should say these agar plates, are clear. What we would have to do, and if we were in lab right now, we would be doing this. I would send you out into the environment around campus. You would have a cotton swab, and you would kind of scrape, kind of just with the swab, a moistened swab, scrape anything that you'd be interested in analyzing to see if bacteria was growing on there, right? So we gently swab, and then we bring the swab and we kind of streak it across our plate, right? So we just kind of streak it. Normally we do it in a side-to-side motion. This looks just kind of randomized, but we would kind of streak back and forth, back and forth, back and forth. After we do that, we incubate them at body temperature for, I don't know, a day, 2 days, depending on how much time we have. And then we would come back and analyze. And what you see here now is a plate, petri dish with bacterial cultures. And we call these little circles, we call those plaques. I don't know if you've heard of the term plaque that gets on the teeth. A plaque is not just one bacteria. A plaque is millions of bacteria. It's a whole colony of bacteria, right? So the very first type of analysis we can do is, well, was there bacteria on that object or not? So if nothing grew, if we have no plaques, well, we say, no, there was no bacteria initially that were sort of introduced into our plate. Two, if there was bacteria, then we can do a visual analysis. We can see the different types of colors. So there's some green colony, a green plaque, a red plaque. We have some whitish plaques, yellowish plaques. And then we have these little things here, these little green fuzzy things, right? That gives us an idea that we're not just dealing with bacteria, that's actually fungal. So that's a fungus. So it would give the doctor or the researcher an idea of what types of things they're dealing with there, right? So it's an important first process, right? First part of the analysis, first part of determining the species or type of bacteria that we're working with. The plates, the culturing, the incubation, and then the plaque analysis, right? So once we have our plaques, here, we can kind of go in there, sort of extract a little bit of maybe this whitish plaque or maybe some of the green plaques, right? And we can extract them, put them on a microscope slide, and perform a gram stain test, right? I'm going to get into the details of the gram stain test in a bit. But the gram stain test will then allow us to see the type of cell shape. So if you're one cell, if you're a prokaryotic cell, you're single-celled, one cell, there's a very sort of limited amount of variation that that one cell can acquire, can attain. Right? So we're going to break it up into 3 types of shapes, right? So one single cell could be sort of oval shape. One single cell could be round, or one single cell can kind of be snake-like and spirally kind of. So that doesn't sound very scientific. Round, oval, spiral, right? So the taxonomy tries to make it sound a little bit more complex. We go now to the Greek language, to the Latin languages, and the terms that is given now, if the bacteria are oval-shaped, we say they are bacilli. If the bacteria are round, we say they are cosi. If they're spiral, then we term them as spirochetes. So I want you to practice that vocabulary. Cosi, bacilli, and spirochetes. So these are different cell shapes that we can start to address. You're going to hear some other terms. You're going to hear the word staphylo, right? So staphylo is the term that implies a group. So if I hear the term staphylococi or staphylococcus, coccus meaning one, coci meaning many. So staphylo means a colony, a cluster. Coci means round. So staphylococcus would be a bunch of round little clusters together. If you hear the word strepto, we heard that before, strepto. So strepto would be like a little chain, right? So we have one connected to the other, connected to the other, connected to the other, like a little line of sausages, of hot dogs. If we're talking about a streptobacillus, right? It would be a chain of these little individual cells. So just some vocabulary that you might hear. So bacilli, coci, spirochete, and then the cluster that we call staphylo, or the linked chain that we call strepto. Right? So different vocabulary that I'm going to be using here. So we look at the idea. This is an example of how we would be able to see these under a microscope. So you would be able to see a really sort of small roundish bacteria that would fall under the coccus designation. So this species is called Enterococcus. So you hear the word coccus in the name, oh, well, that's got to be round, yeah? Entero, it means sort of internal. So that would be an internal round bacteria. So there is some information held, it's not in the English language, but there's information held in these scientific names here. So bacillus would be then these rod shapes, oval. Very common bacteria may know of E. coli, Escherichia coli. It is a bacillus-shaped bacteria. And then we have a spirochete. We have a spiral bacteria that lives in the water. So we call that aquaspirillum. Right? So just giving you a little insight as to how these names come about. The names do make sense if we speak the Latin or the Greek languages. If you just speak English, these are just crazy sounding names without a lot of, any context to them. Right? So, but again, just trying to emphasize the names coccus, bacillus, and spirochete. Plural, coci, bacilli, and spirochetes. So maybe in the lab, they might have you do this. So maybe showing pictures of the different cell shapes and then you identifying, right? So what would you consider this top right? I hope you said bacillus, bacilli. Left, I hope you said coccus or coci. And bottom right, I hope you said the spirochete. Right? All right, so, okay, we've found the cell. We're analyzing it. We're able then to see the cell shape. So we can start to see the cell shapes now. Now, another analysis that we're going to do is the analysis of the cell wall, right? This is a very important molecule. I promise you questions on this for the next exam. The identification of this protective molecule in the cell wall that we call peptidoglycan. And so peptidoglycan is a combination of a peptide, so proteins, and sugars, right? So peptidoprotein, glycan referring to sugar. So peptidoglycan, a protein and sugar molecule that makes up the protective cell wall around the bacteria. So this picture should mean something to you, right? So we're looking at the cytoplasm of the cell, and the cytoplasm would be here. This would represent our phospholipid bilayer, right? The hydrophilic, hydrophobic, hydrophobic, hydrophilic, phosphate heads, fatty acid tails, integral membrane proteins. So you should know this from 1306, right? Old stuff. Well, the new information here would be then the cell wall. So this combination of proteins linked with carbohydrates, very thick protective layer that occurs external, outside of the membrane. And this molecule with proteins and carbs, we would call peptidoglycan. So let's say we're trying to destroy a harmful bacterial species. We got to get in here. But before we can get in here, we got to break through all that thick layer of protective cell wall, the peptidoglycan layer. So again, peptidoglycan is an important molecule to know. It's the strength, it's the protective layer of bacteria. And if we want to destroy the bacteria, we got to know the bacterial's composition, the cell wall, maybe any weaknesses that we can find there. So a very important molecule, peptidoglycan. So I mentioned the Gram stain test. So when we look at the Gram stain test, basically, this is an old procedure, right? This was in the late 1800s, right? So over 100 years old. When we say gram doesn't refer to a mass, not the weight of something, right? We're looking at gram as the name of the scientist that discovered this. So the Hans Gram, discovered a test where he had different kinds of stains, different kind of like sort of like food colorings, right? Different staining liquids. So it's a very primitive test, right? From the 1800s, but we still use it today. Right? Why do we still use it today since it's so old? Well, it's simple and it works. It gives us the information that we need it to provide us. So the Gram stain test is going to involve 4 liquids, right? One, we're going to have a crystal violet stain, some iodine, alcohol, and then a saffron red stain. So the violet stain and the red stain are just stains. They're just different types of purple or red stain to give, you know, a little bit of color so we can see boundaries, we can see edges, so light is not just passing right through these little bacterial cells. Get some contrast with these stains. The iodine is going to be important because if we're working with bacteria, maybe it is pathogenic. We don't want to be working with active or live bacteria. So the iodine would be useful in the killing. We're going to heat fix it. We're going to use heat as well, but if you need to survive the heating process, then the iodine will be important in killing them. And then also alcohol. So whatever the iodine doesn't kill, the alcohol will kill, and the alcohol also is good at extracting fluids from these bacteria and extracting excess stain from these from the first process, right? So I'm not going to get too involved with the Gram stain. I think you're going to get that experience in the lab. So here we have the violet used first, iodine, alcohol, and saffron red. Right? So after we're done with this, it's not a long process. It's pretty quick. When we're done, we're going to get 2 options, right? We're going to get a result that we call Gram positive or a result that we call Gram negative. So that's the 2 options that can happen from a gram stain test, right? Gram positive result or gram negative. So now let's look into what do these results mean, right? So what if it's gram positive? So what if it's gram negative, right? So let's begin with the idea of a gram positive bacteria. So if we're looking at a gram positive, visually we're gonna be able to tell because we're gonna see a purple stain, right? So after we do all 4 chemicals, the bacteria will appear purple. So if it's purple, think of purple positive, purple positive. So it will be a gram positive bacteria. Now this is significant because gram positive, purple staining bacteria, gram positive bacteria will have a very thick cell wall. So that's important because, again, with a very thick cell wall, we know we're going to have to work hard to break through that big, thick protective barrier. Right? So it's just big, thick cell wall and nothing else. So that's important. To make matters a little bit more complicated, some of the gram-positive bacteria are capable of releasing exotoxins. So let's say this, I don't know if you've heard of anthrax. Bacillus anthracis is the bacteria that causes anthrax. So Bacillus anthracis is a gram-positive, purple staining, thick peptidoglycan cell-walled bacteria. When this bacteria is just normally metabolizing, it's just normally growing, normally metabolizing, it generates wastes and it starts to excrete these wastes. So it throws the wastes out. So we call exo out. So it throws the waste out of its cell. Now, if this cell is in the human body, it's throwing the waste out, but those wastes then now enter into the bloodstream of the human. And again, exotoxins are very potent protein molecules that our body reacts very harshly against. So they are very symptomatic. They can cause pain. They can cause different types of medical conditions. So again, not every gram-positive bacteria releases potent toxins, but every gram-positive bacteria does release protein molecules. Right? Some are not very pathogenic. Some are very pathogenic. So there's 2 gram-positive bacteria that I would like for you to know, right? The anthrax, and then a family of bacteria that we call Clostridium. Right? So Clostridium, you may have heard of Clostridium tetani. You may have heard of tetanus, right? You step on a rusty nail, you're at risk of getting tetanus. Well, tetanus is one of these gram-positive bacteria that if that bacteria gets in the system and starts releasing these exotoxins, or if you come into contact with the exotoxins from an outside source, these toxins cause the muscles to start contracting. So they cause the muscles to enter into a state of tetanus. I don't know if you understand what that means. In the medical vocabulary, tetanus is a severe, sustained, very strong involuntary muscle contraction. So you know that basically as a cramp. So you've been asleep, you wake up with the pain is that your arch or your calf or some muscle is starting to cramp, right? So we know cramps are painful, right? So imagine we have then systemic body-wide cramping. That would be very, very uncomfortable, right? And our body reacts harshly to these gram-positive proteins, these exotoxins. This is a picture of the 1700s. An artist drew a patient suffering from tetanus. So you can see here, starting at the legs, the feet, you see the arches that are cramped, right? The toes have crunched inwards. We have cramping of the calves, cramping of the hamstring, of the gluteus, of the lower back, of the biceps, of the hands, the neck. So again, you can imagine how miserable this would feel, right? So in terms of being fatal, I don't necessarily know, or I wouldn't necessarily claim that it's that fatal, but it must be excruciatingly painful, and again, just something that we wouldn't want to endure. So in the United States, we have great respect for tetanus. So we basically make it mandatory from a very young age for children to be vaccinated against this. So in other countries, they don't vaccinate against tetanus, and children then have to endure this terrible condition for them to build immunity. So in the US, we say, man, we don't want the kids to go through that, so we vaccinate them. They get the immunity without having to undergo the harsh symptoms. So some other sort of, I guess, symptoms, the lockjaw. So it starts to affect the masseter muscle, the jaw muscle. So that muscle's cramped as well. So you can imagine your teeth just sort of cramped, shut together. You know, it's kind of sad to see these young children suffering from these what must be very painful types of symptoms. So again, this is why we have great respect for gram-positive bacteria. To complicate things, right? So gram-positive bacteria have a lot of peptidoglycan, and they're able to then form endospores, right? They're releasing exotoxins, but in certain situations, they can form endospores. Endo refers to inside. So let's say that you have a gram-positive infection, and you go and you get the correct antibiotics. Antibiotics are attacking the peptidoglycan. So we're going to attack the peptidoglycan. And once that peptidoglycan starts to get weakened, this bacteria is sensing it's alive, it's a living thing. It senses the world around it, and it decides, man, this is not looking good. My genetic material is at risk. If I do nothing, they're going to break through this peptidoglycan. Now, different strategies may come to mind. Well, if I'm a bacteria, you think, well, let me go back and start trying to repair the damaged peptidoglycan. But there may be attacks around the entire, three-dimensional, around the entire cell. So that may be a lot of work. So what the bacteria does instead, it starts to make peptidoglycan around the DNA, inside of the cytoplasm. It makes a cell wall that's supposed to be on the outside, but this cell wall is now inside. We don't call it a cell wall, we call it an endospore. But it's basically a cell wall on the inside of the cell protecting the genetic material, the DNA. Once the bacteria does this, it kind of goes dormant. It kind of goes to sleep. So if it's asleep, it's not metabolizing, it's not releasing exotoxins, you are going to start to feel better. Now, here's where problems arise. I'm going to pick on some of you here, right? Some of you, I bet, have half-used bottles of antibiotics at home. So that tells me you don't follow directions. The doctor said, be sure and take all of the medication. Take the full bottle. Maybe you have enough pills for 10 days. But after maybe 5-6 days, you start to feel better. Oh, I feel pretty good. I have some extras. I'm going to save them for next time. Well, what may have happened was that you started to attack, you started to break through that peptidoglycan. The bacteria started to form endospores, went dormant, and you stopped taking the antibiotics. So that means you didn't quite destroy the bacteria. You just made the bacteria go into hiding. Okay? And then you go out, you live your life. After a period of time, could be days, could be weeks, could be months, the bacteria sort of like a little seed, it kind of germinates again. And then you start to have the same types of symptoms, right? So you didn't kill it right the first time. And we talk about now evolution. So you may have heard that little phrase, that which does not kill you only makes you stronger. Well, you didn't kill the bacteria, but you gave the bacteria a sense of what type of antibiotics are out there, and the bacteria then can now modify and adapt and maybe mutate. So that can lead then to bacterial resistance, stronger strains of bacteria. So please, 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 if you use antibiotics, follow the doctor's orders, take the correct antibiotic, and take the full duration of the antibiotics. Right? Those are important. So again, just to summarize, gram-positive bacteria would have stained purple. So this would be dark, dark purple. And if it starts to be attacked, we start to see this peptidoglycan now on the inside down here of the cell in the endospore. If the bacteria is just left alone and it's secreting the exotoxins out, exotoxins would kind of seep through here into your bloodstream and cause you to become sick, possibly. All right, so here's a like a picture showing then the gram stain result. So we see now the purple coloration. So purple on the peptidoglycan thick cell wall. You can see where that's being attacked, thick, thin, thin, thick. So we have different thicknesses of the cell wall. That's kind of showing us that the cell wall is beat up, it's damaged, it's being, something's trying to break through. How did the cell react? Well, that bacteria started to make little endospores, protecting little fragments of DNA inside of the cell. So this bacteria at this point is not dead, but it is dormant. It's just waiting for the right conditions to regerminate, basically. So that's the gram positive. Now let's contrast that with the gram negative bacteria. So gram negative would stain red to pink. And gram negative is going to have a very, very thin cell wall. So big distinction, very thin cell wall, not very well protected. However, external outside of the peptidoglycan, thin peptidoglycan cell wall, we're going to have another layer that we call the lipopolysaccharide layer. Lipopolysaccharide, or LPS. Sometimes you hear it called LPS. So most of the antibiotics are going to target peptidoglycan. So we don't have a lot of peptidoglycan in this type of bacteria. So it's a very minimal defense. But what it does, it tries to hide. It covers all the way around with LPS to try to mask, camouflage, hide then the peptidoglycan layer. Right? So it's a sneaky strategy. These bacteria are not considered as potent as the exotoxins. So the medical community doesn't really concern themselves too much with gram negatives. There are some exceptions, but overall, if you have a gram negative bacteria, you can expect these three classical symptoms together, right? Like a low-grade fever, vomiting, and diarrhea. So these two together, vomiting and diarrhea, we typically associate with like food poisoning. I talked about salmonella in the prior discussion. Salmonella, E. coli, these are gram-negative bacteria, and we can associate these symptoms when we have these types of infections. You're not going to die of salmonella. You're not going to die of E. coli. You're going to be miserable for a while, but as long as you can stay properly hydrated, keep the fever, in check, you're going to survive, right? Now, interesting part of the story. When do you become sick from a gram-negative bacteria? You become sick when the bacteria itself has become damaged or lysed. So as long as the bacteria is intact, like right now you may have E. coli in your system and you're fine, right? Because the E. coli is intact. Once E. coli is damaged and is broken, then it releases the components on the inside of the LPS. So they're inside of the LPS, and then they are released out into the body. So in contrast, going back, gram-positive bacteria cause you to get sick when they're just normally metabolizing. They're alive and healthy. Gram-negative bacteria are going to cause you to get sick when medication or your own immune system is destroying the bacteria. So we kill them, the bacteria is dying, you killed me, but I'm going to give you diarrhea, right? It's that last little sort of evil that it does as you're killing this bacteria. So this picture shows a bacteria, a bacillus form of gram-negative with all its DNA scattered out here. It's like in Halloween or scary movies, maybe you have a body and all the intestines are laying outside the body. That's not good, right? So that person's going to die. This bacteria is going to die. So all of the DNA is kind of scattered out. That bacteria is dying, and as it's dying, it's releasing fragments of LPS, causing then these classical gram-negative symptoms. So I want you to contrast this with the previous picture of the gram-positive, right? So here we have the cytoplasm. We have our membrane, very, very thin peptidoglycan layer. And then external, surrounding in 3 dimensions, now we have our LPS layer. It's basically another membrane, right? It's basically like similar to the plasma membrane. It has a different composition than this one. But if I'm an antibiotic, I'm up here and I'm looking for the protein, but I'm not going to find the peptidoglycan protein because it's hidden internally. So again, this is where different antibiotics come to play. This is where different effects on the immune system are factored in. It's just a different strategy, trying to protect the cell with only a thin layer of peptidoglycan. So for very, very different behavior. So just to kind of show you how this would look in real life. So we get our plates, we culture our plates, we incubate, we analyze the plaques, we extract some little cells, put them on a microscope slide, perform the gram stain, and this is what we would see. Right? So you would then now be able to distinguish maybe some round, some foci, maybe some ovals, some bacilli. I can see some pink. I can see some purple. So this would give the doctor an idea, oh, this is maybe 2 types of bacteria. We're looking at a gram-positive and a gram-negative, or we're looking at bacilli and foci. So this is, again, important first step information to determine the species of bacteria. So when we look here, we're looking at the much more dense population of bacteria. And then we bring in that term again, staphylo, right? So this is actually from the mouth of a toddler. So a young child, they never brush their teeth. They're always eating stuff. All this bacteria growing in there, right? But we can see now gram-positive, little cluster of circles. So we would call this staphylococcus, gram-positive staphylococcus, and then gram-negative bacilli. So it's just kind of individually scattered. So looking at 2 different types of bacteria in this situation here. So this is how this information would be used. So again, this is a very good summary slide. Learn this slide, spend some time with this slide. I promise you many questions based on this. Compare, contrast, gram-positive from gram-negative, right? Gram-positive being purple, gram-negative being red to pink. Gram-positive, thick layer of peptidoglycan and only peptidoglycan. Gram-negative, very thin layer of peptidoglycan. And we have an LPS layer, right? External to the peptidoglycan. So again, thick peptidoglycan, very thin peptidoglycan. We have membrane, peptidoglycan, and LPS for gram-negative. We have membrane and only peptidoglycan for gram-positive. Gram-positive would release exotoxins when healthy, when normally metabolizing. Gram-negative would release endotoxins, this lower level of the LPS, when that membrane has been damaged and ruptured and torn. Gram-positive capable of making endospores. Gram-negative does not have enough of the basically enough of the peptidoglycan to make these endospores. Right? So important compare and contrast with all of these types of little details, aspects, that kind of stuff. So if we were in a microbiology class, we would continue learning more of the taxonomy, more individual species. We're not going to get that far involved in the 1307 course. I did mention this a little bit, just want to reiterate a bit. Bacteria are becoming more and more difficult to kill. And what do we blame for this, right? Well, we blame the idea of sort of overexposure to antibiotics, not taking the right antibiotic, not taking it for the right duration of time, taking the incorrect antibiotic for the bacteria that you actually have. So all of this is helping the bacteria to mutate, the process of natural selection, right? We're killing off the weak ones, so who survives? The stronger ones. Right? So bacterial resistance is becoming more and more and more and more of a problem, right? So I don't know if you've heard of MRSA, methicillin-resistant Staphylococcus aureus. It's associated with necrotizing fasciitis, the flesh-eating disease. This bacteria, Staphylococcus aureus, is a very minor type of bacteria. It shouldn't be that bad, but we're having patients going into the hospital and coming out with these very aggressive, very strong strains of bacteria. So the strains are genetic modifications. It's the same species, but they behave very differently than the normal species because of different selective pressures, mutated strands. So again, just please, 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 again, follow doctor's orders. Don't just self-medicate. Take the appropriate antibiotic for the correct duration of time to minimize some of this mess. Here we just have a few of the just maybe common diseases you might have heard of, right? Strep throat, tetanus, syphilis, and then you see some of the species that are associated with those. This is a whole course in and of itself. So just, again, we could spend the whole semester on diseases caused by different types of bacteria. All right, so for now, let me back up here. Let me stop this one. I have, after this, just one more discussion. I'm gonna look at 3 different types of bacteria, and then we'll talk about the archaebacteria. But again, we've dove quite deeply into bacteria here. And that's, I guess, enough right now to keep you on track. And I'll be uploading some more here as soon as I can.